Hello, John. It's so great to see you. Thank you so much for having this conversation today.
And thank you, Andrea, for inviting me. It's first we emailed back and forth, but the first time I've seen you live, I've seen your podcasts and things
Yes,
like
that's
that.
true. Patricia Churchland introduced us way back, but we are only finally getting to meet in person. Although I have read a lot of your papers over the years that I want to share with others, but let's just start with the biography. And before we start, I also want to say we're going to talk about some issues relative to neuroscience, but try to talk about them in a way that's a little bit general and try to find ways to really think about and explain these topics in a way that a lot of people will understand. So, but first, John, I think it started with snakes and smell. Is that right? Do I remember
That's
correctly?
pretty early. That's not quite the start,
Okay.
but I mean, just I'm an older guy, and when I was in college, neuroscience wasn't such a thing, and especially we might call cognitive neuroscience was in its infancy or hadn't been born yet. Whatever, it's exploded in recent years. And in college, I was a biology major, but I was really interested in psychology. Actually, I had a great uncle who was a psychiatrist who was very influential, but I didn't really know that there was any fusion of the two fields. And it was actually 1 year out of college that suddenly, aha, brilliant. You can do both a little bit. And I started a graduate program.
Do you remember what the aha was?
I was working actually in a pediatric neurology clinic,
Oh,
helping out. And I took a part-time, took one course, audited by a professor in the program, Walter Riss, and it was called Comparative Neurology. And it was a comparative description of brains. And one of the things that excited me was that it had a point of view. He presented, and I did science, taught with a point of view. That's really great. That's exciting. And he was really a brilliant guy, very interesting lecturer. So
what was exciting about that? The point of view. Did it mean you could? Yeah.
That it wasn't just balancing all the facts. You could start with an idea and present, to defend it as strongly as you could, that kind of thing. He had a few basic theses that he would present throughout this introductory course. One was levels of organization within the CNS, that basically, you had a theme and you could build them out in different animal brains. And actually, he was the original editor of Brain Behavior and Evolution. And so that's the core of the interest. So when I started graduate school, it was in his group of faculty. It was in the early 1970s when neuroanatomy and comparative neuroanatomy was a much bigger component of neuroscience. And my advisor was in the group, a woman named Mimi Halpern, and she was studying snake brain, and she was more of a psychologist than she was learning to be a neuroscientist and neuroanatomist. And she was interested in studying the two olfactory systems in snakes. And just a year or two before, others in the group, Frank Scally and Sarah Winans, had shown that the central connections of the main and the accessory olfactory bulbs in mammals didn't overlap. And the accessory bulb is the relay for the vomeronasal system. So this really reopened the idea that these two olfactory systems had different functions. They could be viewed. They weren't just redundant or overlapping in some way, which was sort of the preponderant idea. But anyway, so it turns out that snakes have a fantastically and relatively enlarged vomeronasal system. When they flick their tongues, what they're doing is picking up chemicals from the environment and delivering it to their vomeronasal organs, which are in the roof of open into the roof of the mouth instead of into the nasal cavity. The epithelium is very similar to the olfactory epithelium, but it's a second sensory system. And there was some rough idea that maybe this second system, this vomeronasal system, is sensitive to non-volatiles and non-airborne odorants. The tongue would be a good way of touching and picking them up and delivering them.
Oh, that's so fascinating.
So
I love going to natural history museums where you can actually see, for example, I'm thinking of the dog because I have a dog that I just walked. And of course, the sense of smell is so prominent. The way that different creatures sense the world through their smell is really fascinating. But were you actually interested in that when you were young? Yeah. Okay.
No, when I was young, no. It all came through this, starting graduate school. And when I started graduate school, I wasn't at all sure that it could be a full-time scientist.
Oh,
the idea of spending hours a day, 12, full time doing stuff. I've been and worked in labs, and it wasn't, college laboratories, which last 2 or 3 hours, are excruciating, high school
yeah,
especially.
that's what I'm trying to get at. What was like driving the interest? Because you got very much into technology, for example. I mean, you're doing science in the lab. You're good with the technology. Were you interested in that stuff when you were a kid, or was it more curiosity? I know you were interested in emotions at this time. At least I think
Well,
I
it's a mix
remember
of
correctly.
things. So compared to my family, I'm very good with my hands, but
Okay.
that's a bad comparison. I remember, in fact, science toys in the 50s were really great. I had a whole bunch that were, not just microscopes and Rector sets and things like that. I remember I had an analog computer set. I didn't quite know what it was, but I put it together, tried
Oh,
to
wow.
get it to some math and things. Anyway, so going in the emotion side probably is similar to all of us. We're all a little mixed up, and we want to know, where that comes from, if we can fix that up a little bit. And I did have my great uncle, Lawrence Kubie, was a renowned psychiatrist. And
Oh,
he was
wow.
the sort of the famous person in our family. So
Oh, wow.
he was very motivated. When I went to college, I went to Johns Hopkins, and he lived just outside of Baltimore. And I visited him a lot, and he was really inspiring. It was a lot of fun, but also, sort of helped me focus a bit, although not finally. But those
Yeah.
are some of the
I
influences.
can imagine that would be influential on different levels. I mean, when you hear your family talking about him as a famous man, that's influential, but also when you come to that age where we all go through, whether it's adolescence or later, where you are, like, as you said, you feel a little confused, you're trying to figure out the world. It seems like it would then kind of, yeah, that would fit naturally as a place to look, maybe psychology, if you had it already in your environment or context.
Yeah, and I think I was introspective, so that would, fit in with that sorts of things. I would say that.
But how
But
does
jumping,
snakes and emotion and all of this come together? And, at some point we go to the hippocampus. So this seems like these are far away, but were they?
let me just, be contrarian a little bit.
Okay.
And McLean's triune brain. Is
Uh,
that familiar?
I do, but we should talk about it. The limbic and
Yeah, so
yeah.
Paul McLean was an important historical figure in introducing actually the study of emotion into neuroscience.
Hmm.
And he, I think, coined the phrase the limbic system.
Right. Which
And
everyone says we shouldn't use anymore. I don't know how you feel about that.
I use it.
Okay.
I use it in a way that's not very controversial. I say it's an anatomical system. It's defined by its relationship to the hypothalamus and not in its functional
Yeah.
role. So Paul McLean, the famous triune brain, and he's criticized for roughly saying there's a reptilian brain, and then on top of that there's a proto-mammalian brain, and on top of that is a neo-mammalian, as if these layers spring up from nowhere. And every biologist, including Paul, knows that nothing comes from nowhere. Everything derives from something. And Paul McLean knew that well. There's one just terrible figure that he used, everyone used, that seems to show these layers as if they just one grew miraculously, like, from the head of Athena on top of another. And that's just silliness. And he never really said that. And I've reread his book, The Triune Brain. It's not a very good book, but his actual work was much, much better than that. And he had a pretty sophisticated.
It's one of those things too that just went into popular culture as people talking about the lizard brain, as if we've evolved from the lizard brain. And, in the past 5 or 6 years, how emotions are made, the book has come out and it's kind of saying, okay, that's a really wrong way to think about emotions. And so
Yeah, I think that's a really wrong way to dismiss emotions. I think we did evolve from something like an ancestor to lizards, and we don't understand it very well, but that's a real wonderful problem to explore.
Well, you studied salamanders and snakes, so you probably see a different connection than I'm able to really see. I mean, do you see some kind of continuity in a more general sense?
Yes and no.
Okay.
So I think the biggest unknown let's back off a little bit. So there's a wonderful book came out a couple of years, Strider and Northcutt, called what's the title? But it's The Evolution of the Vertebrate Brain. And they're principally comparative neuroanatomists and very detailed in comparative neuroanatomy with some jumps into function. They're not great in physiology. It's an example of such a broad area that nobody can know everything. it's a very good book. But among all the books, the big mystery is where neocortex comes from. Because neocortex, we all know, is really important. And its roots are not agreed upon. And even if they were agreed upon, if we know that, say, it came from dorsal cortex, it almost certainly functionally very different from it's doing, the algorithms, whatever it's doing is really unique and different. And so I don't think there is a great idea where this wonderful neocortex came from. And connected to that is, well, this wonderful thing evolved. What's it doing for us? What benefits do mammals have that have a neocortex that reptiles don't have, and birds have another version of something that does something sort of like neocortex, but it's not a neocortex? These are really interesting questions, and you have to get into the functional processing level, which is just beginning to be understood, to get at that. And I think that's a really wonderful avenue to get at that's barely scratched the surface.
Might be a good place for people going into.
Yes, there are a couple of labs working in that area, and just a couple, and I'm not, the name's not coming to mind, but it's a very rich, I think, a rich area. It's, the rewards are huge if
So when you were working with the snakes and smell, what were you working with? What was like the kind of technology or the equipment or the procedures, methods?
One of the problems with my thesis work was, I love my thesis work.
Your PhD was on that.
Yeah, 7 papers came out of it. There's really, it's one of the things where we simply ask questions and answer them. They don't have to be investigated again, because it was such a new field. You could just say, is the Vomeronasal system critical for mating behavior in male garter snakes? The answer is yes. It's an all or none. It's not like,
that sounds so satisfying. Rarely do you get such satisfaction in science so quickly.
And we did develop some novel techniques, but they were on the behavioral lines. So Mimi was very good at doing the surgery and very carefully cutting the olfactory or the vomeronasal nerves. We had great histology to verify the lesions, and we worked in understanding the histology and degeneration of the organ systems. I developed a technique, suturing closed the vomeronasal duct so the animals couldn't deliver tongue flick odorants into their vomeronasal organs. And we anesthetized, we did local anesthesia, injections under the chin, so caused paralysis of the tongue muscles, and they couldn't flick their tongues. As soon as that returned, all the behavior, the moment it returned, the behavior returned. Wow. That kind of thing. So a lot of video analysis of tongue flicking, sort of primitive, but there was very little background to work with. We were free to develop crude but interesting techniques, and it was a lot of fun. And
fascinating PhD, but then your postdoc was...
Well, so it was fascinating, but I didn't learn any deep skills. That I knew I wanted to work in the limbic system. I think I wanted to work in mammals. I wanted to do electrophysiology, and I had no idea how to do electrophysiology. So the first postdoc I did was at Penn with a guy named David Moulton, recording directly from the olfactory epithelium. So using electrical recording the neurons in the brain use electricity. The signals they send from one neuron to another are electrical signals. Action potentials are in the other signals. And so with electrodes of various sorts, you can put the electrodes either in a gross area and get gross waves like EEGs and field potentials, or with a fine-tipped electrode, you can record, say, outside of a single neuron and listen to the activity of that single neuron. Action potentials go up. Or something I haven't done, you can use even a finer tipped electrode and go punk into the cell and record the electrical activity within the cell that leads to the cell having an action potential or not. That is, you can listen to its inputs and see how they add together. And so there are a whole variety of ways of using electrical recording techniques to probe the brain. And
that was all being developed, right? As you were going to school?
Yeah, this is right. So 1977, I went to work at Penn.
Late 70s then, okay.
Right, this was not new. I'll get to what's new. So I was just, the recording from the olfactory epithelium and salamanders was relatively straightforward, simple things. We were recording what's called an electro-olfactogram, which is putting the electrodes on the mucus layer of the epithelium.
Okay.
And recording a masked response of many neurons when an odor is delivered. And these are pretty big responses. And the salamanders, we just exposed the epithelium, and we were able to make maps of sensitivity in different parts of the epithelium. And
when you say maps, you could
say the anterior part of the epithelium, the set of neurons there was more sensitive to water-soluble odorants, and the posterior regions to less soluble odorants. And we mapped out different odorants and their, where they would have peak electroolfactograms, and the generalization was that. So that's what I meant by a map. It wasn't a map in terms of hippocampal maps or navigation, but as I said earlier, I really wanted to get into the brain, into this emotional stuff, into cognition, which we may get back to. And this was not cognition, but it taught me a lot. So in terms of new techniques, so I went back to my PhD school, down to the medical school where I am now. And Jim Ronk had just moved there from the University of Michigan. And we'll go into Jim, I think, in some detail, I hope.
Yeah, so we're at late 70s, right?
This is 79.
This is your second postdoc, and you've got the experience recording a bit. And Jim, now you go to downstate for the first time.
Right.
In Brooklyn.
And I had planned to go back and work with Jim Ronk. He was an exciting guy. And among the exciting thing that he did was be on the forefront of being able to record single neurons in behaving animals. So we go back to that electrical recording technique. There's a pretty small electrode put into the rat's brain, and it's not going into a cell, it's getting close to a cell so it can listen to the activity of hopefully single cells. So this was technically very challenging.
So you're listening to the action potentials.
Right. But so it turns out that Jim Ronk and John O'Keefe were two of the pioneers in being able to record single neurons while the animal's walking around in space. The biggest problem is
O'Keefe was at McGill. We kind of already talked about him a little bit with Len, and he was just starting to use the same technology that you were using with Ronk at the same time. Is that fair?
Yes and no. He was at University College London. He left McGill.
Oh yeah, right.
He
was already at UCL.
And he was recording from the hippocampus, and Jim was recording from the hippocampus. They didn't know each other. The important technical advance was to use something called the FET, or a field effect transistor, which put a lot of up the signal going up the recording cable. The action potentials of single neurons are just tiny amounts of electricity. And if you tried to put that tiny amount of electricity up a wire, any movement of the wire would make huge artifacts, would induce currents. And so the problem with behaving animals, it's going to walk around, you've got a wire going to its brain, and you get this crazy noise coming in. But the FETs mounted on the head stage amplified the signal so that the electricity going up the wire was much stronger, and meant that the relative electricity induced by moving the wire was really small and could pretty much be ignored. So Jim and O'Keefe were among the pioneers, maybe the pioneers, in the ability to record single cells in behaving animals. And these FETs were brand new, but they were robust enough to permit this, and they were good enough engineers to see the possibility.
And did you know that Jim Ronk was doing this already, or?
Yes.
Okay, so that's why you went there. You wanted to be part of that. Okay.
Right. And I'd heard from Jim, I think, about these things called place cells, that cells fired where the animals wasn't in a place. And
so was this already the 80s then? Because that wasn't that 78 that he found
this is 79.
Okay, so you were really on top of, you were like already reading all of this.
Right. Reading, I don't know. So I heard from Jim, word of mouth. Jim and O'Keefe were working in parallel. They didn't know each other. Which, as Jim says, he heard about O'Keefe, and he wrote him and said, "Can I come over and see what you're doing?"
Right.
And he spent part of the sabbatical over there, got to know O'Keefe and Lynn. And as I remember, he didn't see any place cells because they weren't recording stuff in the months he was there. But he was he liked them a lot. I don't think he was not 100% convinced, but he was on their side. And
because he hadn't actually seen them. But he had not recorded head direction cells yet.
He had he was recording single neurons, and he has a paper of the behavioral correlates of single neurons, which goes totally different directions. He was not looking at, he was not paying much attention to where the animal was. These things called approach consummate cells or approach, and he'll deny that they're meaningful at all because he just thinks he got it wrong. Although Lynn and John O'Keefe are much more charitable. And
yeah, I've noticed that. I mean, it seems like this time period at Downstate, him and then you and then Mueller, will get into it, seems very important, but when Ronk is asked about it, he seems to kind of give credit away. Is there a reason for that? I mean, we haven't gotten into head direction cells.
I think it's very important, but I don't think that he would say he had anything to do with the discovery of place cells. So yes, he thinks O'Keefe's wonderful. He gives him full credit. I think in the beginning, in the introduction to the cognitive map book, they say Jim sort of saw the same thing. Jim denies it. That kind of thing.
Yeah,
so
I remember that part.
They're mutual admiration society.
Well, that's fascinating. Okay.
And so
you went to work with him, and he put you to work on this 3 behavior task that's sort of similar, right?
Yeah. So in those days, Jim wrote, I think, a 200-page grant proposal. Whoa. And it was all about this 3 behavior. He sent it to me, or I said it was, just this stuff pasted together. It was crazy. But it was about the 3. That's what people did in those days. And if you sent in 300 pages, you got funded, I guess.
So.
Who had the patience to read it? But it was
nobody wants to read it, so just give them the money.
The 3 behavior tasks, and the 3 behaviors he chose were a radial arm maze, which was developed by a guy named David Alton, and it was used early on for lesions and navigation, spatial behavior. Used a lot. It's very good apparatus.
Sort of before the water maze.
Yeah, it was the chief task before the water maze was developed.
Richard Morris, who will also come on the show and talk about everything too.
Right. And the second apparatus was a large maternal box, very big, and with these mother rats and the reason for that is that it was reported, and it's true, that mother rats will have a difficult time retrieving their pups after hippocampal lesion. They'll retrieve them, but they won't put them back in the nest. They'll pick them up somewhere, put them back somewhere else, that kind of thing.
Huh.
And
very sad.
And they perhaps didn't die. I mean, they were still well maintained.
Yeah. Okay.
Anyway,
I'm going to hold my personal feelings in check.
Okay. And the third thing was something called the DRL-16. And DRL is differential reward for low rates of responding. And it meant the animal had to press a bar and wait 16 seconds before pressing again. And then if it waited the full 16 seconds, it would get a reward. And Jim had built lovely Skinner box. Actually, 2 different arms could be activated. And DRL-16s also were reported to be sensitive to hippocampal damage. So these are 3 very different kinds of behaviors. Maybe we'll see things uniting these 3 behaviors that will tell us more about the hippocampus.
And for people who have no idea what you just said, because that was hard. The idea was the hippocampal lesions would affect each behavior selected. Is that the selected behavior? Or what was kind of the big idea of the proposal?
The lesion literature showed that damage to the hippocampus disrupted all 3 of these things. There was no mechanistic idea of what would combine the 3 things. It was just, let's take a broad view and take a look at what happens. Do single neurons tell us anything about the relationships of these 3 tasks? And there was no recording that I know of or anything. There were recordings on the radial arm maze, but not the other 2. So what would you see in single neuron recordings in the other 2 behaviors? So it was just exploratory.
So you were the one doing
and so
I
came to the lab and he said, he put me this big, this 200-page grant, he put me on it. He said, you do it now. That's your job. And I was learning all these, how to make the electrodes and
which is exactly what you wanted, right? I mean, you got it. So you didn't really choose the hippocampus, but you chose more the method.
No, I think my idea was I would learn the techniques in the hippocampus and then move to emotion, amygdala, wherever the emotion nexus was going to be. That was
It's funny because I think O'Keefe was doing something with the amygdala, right? And then accidentally
I never quite
found.
figured out what from your talk with Lynn, I got a sense of what Lynn found out about the hippocampus and emotion, or not
Mhm.
emotion, but things, but never knew it. So I was put in these three things, and it was a lot of work to get them all working. Most of the tasks had been built. The recordings were pretty sophisticated. We recorded everything on videotape, with the behavior on
Oh,
videotape.
wow. Yeah, so
And
you had a whole video archive of that. Wow.
yeah,
I
and
guess
the
you still do,
spikes
right?
were
Somewhere.
on the. I'd love to know where they all are, but I'm not 100% sure. So the spikes were on the audio channel, so they were well recorded. In principle, we could go back and analyze it, but we'd never really figured out, once we've collected the data, how we're going to analyze it. So
Gosh, somebody should look at that again. I mean, I won't go off on that tangent, but there could be a lot there. But
right.
So you're recording neurons in the hippocampus
In these three tasks.
in these three tasks, which are different according to what's lesioned, right?
Yeah, they all get messed up with hippocampal damage.
Right.
So the
And what
idea
did you
is,
find?
so what we found was place cells and place cells.
Wow.
That's the only thing we found. In all
And
three
can you,
tasks,
for someone who doesn't know what that means,
okay,
what
yeah,
does it mean that
so-
you found place cells, and place cells?
So what a place cell is, it's a single, again, we're recording from single neurons in the hippocampus. We're just listening to the firing of the cell.
So the action
Ideally-
potentials in the cells, you
Right,
hear
pop,
it. It's
pop,
almost
pop,
just
pop,
like you're just
pop,
hearing
pop,
little
pops.
Geiger counter kind of noises.
Yes, just like a Geiger counter. That's a good analogy. So, and I'll use that in a moment.
I think I got that from you, actually.
And so but ideally, the recording has no effect on the animal's behavior. It doesn't care. It's got some, it does probably, shouldn't care that there's something sticking out of its head, connected to a cable and stuff. It's just running around. In fact, the behavior that we subsequently used, well, in these three tasks, the animal was motivated for different things. In the Skinner box, it was to getting food reward, and they would behave pretty well for that. In the maternal box, the mother was retrieving pups. In this case, normally, because it wasn't damaged to hippocampus. In the radial arm maze, the animal had to solve the spatial problem of going down each arm, remembering which, and finding food at the end, remembering which arm had already been down, and so ideally sampling each of the 8 arms before repeating anything. And that's why it was a pretty good test.
Now, had you read Tolman at this point?
Partially, yeah,
Okay.
quickly, yeah.
So you were aware of this idea of a cognitive map and so on. And I guess that was right when Lynn and John were publishing their book.
Yes, getting back to Lynn and John and Jim Ronk, I think Lynn mentioned that they sent out copies of the book to many people.
Yeah, he does.
And so Jim had a copy that I read.
Wow.
So that would be before I came to the lab.
Oh,
and,
wow. So you were one of the first readers.
I don't remember much about my reaction, but Jim made lots of notes and sent it back to them and stuff.
Is Jim interested in philosophy at all and all these kind of bigger ideas?
Yes.
Or
Jim's, a humanist, very broadly educated. And I would go to Jim with questions about, especially the history of religion. He's really good at philosophy.
Wow.
He was a philosophy major, I think, in college. Jim's, a mentor in many ways, and just, a great guy to talk to, and just, warm and giving and, thoughtful and just, a wonderful inspiration.
Yeah, people always say good things about him.
Yeah, that's true. It's undeniable. I never heard a bad thing. Too modest.
So you got to work with him on your second postdoc, being one of the first people recording place cells in a way, right?
Yeah.
Yeah.
So what's a place cell? So you're recording, you've got this, I'll describe it in a more current task where that's in a cylindrical enclosure, and we're dropping food pellets randomly on the floor. So the rat is maybe food, just searching for food, doesn't know where to go, and occasionally comes a little pellet and eats it. But it's a task that just gets the animal moving. It doesn't have to know where it is in space. It's almost certainly not a navigational task. But while we're recording, we are keeping track of where the animal is. In more recent things, we have an overhead camera that automatically tracks the animal. In these earlier things, we just did video. It was overhead and we kept track of the animal. So imagine that the center of a place field is a piece of radioactive material, and the cells are Geiger counters. So what you hear is, this increased pops from this pseudo Geiger counter when it crosses the middle of a place field or a firing field. And
so there is no It's almost like there's one little area. If it can seem as if you're just watching it, there's some area in this maze or in this area that the rat is where the cells fire only. You hear a lot of crackling and popping, and then elsewhere, not so much.
Yes.
So you're watching an animal move through space, and when it's in a particular area of space, the cell that's being monitored will start making a lot of noise. And when it moves away, less noise. But that's only one neuron. We have to remember.
Right. And so the process I was going through was collecting one or sometimes two neurons at a time with the techniques as they were then. But a lot of these were recording outside of the cells, extracellular. There were a lot of technical challenges, like make sure you're getting the pops, the waveforms of one cell, not its neighbor, that kind of thing.
So this was the first time that someone was understanding what a place cell was. John O'Keefe was the first to figure it out. What is it, though?
If you were thinking from an information processing, it's a cell that tells you, as an outside person, or the rat, where the animal is. The information potentially is giving to other parts of the rat brain or to you as an outside observer.
And that's actually a bit different. So maybe it's working for the rat just immediately, right? That's happening as the rat, and we're modeling it and measuring it happen. But this is just the beginning, right? So these cells were found, you're doing the experiments, and there's many other GPS-like cells.
That we realized that we were recording one neuron out of tens of thousands. Yeah. And we imagined tens of thousands of place cells, although we were recording one or two at a time. And initially, what everyone found, O'Keefe and us and others, is that the cells would cover the accessible area of an apparatus. And that we, at that stage, couldn't record them all at once, but we could see you couldn't predict one or the other. So that was the cognitive map. Single cells are not a cognitive map, but a whole set which gives information to the rat, presumably, or to us, will signal where in the environment the animal is. And
Tolman had kind of had this idea that something like that is happening in the brain, that there's some kind of metric or mapping of the space that is also kind of the knowledge for the rat, but we'll get into that. But this is the very beginning of it. So when you were doing this study, were you thinking, I mean, did you have any idea, like, what were you just business as usual, or were you really thinking like we're onto something really very new here?
Astounding was Nadell and O'Keefe and Nadell's book, The Hippocampus as a Cognitive Map. So your publication was 78. What was it?
78. Yeah.
It's astounding. So it was based on not a lot of cells, but this pretty good sized book is written about a theory based on, I don't know, 20 cells, something like that. And they really put a stake in the ground, said, this is what we think it's doing. The title is The Hippocampus as a Cognitive Map.
Yeah, what a brave thing to do.
And it was not universally well received.
No, because no one had, I mean, this seemed a bit crazy. It still can seem very kind of hard to get a grasp on, but at that time you had Tolman, and then suddenly this area of the brain, which we should talk about, that had in the 50s been kind of locked in with memory because of a person named Henry Malias and HM, this hippocampal area, everyone just assumed was about memory. So this was very weird that now O'Keefe and Ronkin, you and this little group of people, and then Lynn and Nadell writing this book with this big title. This was not business as usual in neuroscience.
I mean, the book laid out a goal, a concept, which is still present. 50 years later, whatever, it's still based, largely right. Not right, but it's got the right ideas. And there's still arguments about is place special, and there's just an example of something. Yeah, they're fun arguments, but basically the book is held up. There's a model. It was exciting. They put a model in, modeling, and it was a pretty terrible model, but it was a model. They tried to put how neurons would do this job. Mm-hmm. They only knew about, very few neurons. Not much was known. It was theta and place cells. That's all they put into their model.
Yeah.
So, but the idea.
I think I say it in my second conversation with Lynn. I can't remember, but I do. In a way, too, that book it wasn't the beginning of neuroscience, but putting all that stuff together. They start with philosophy. You actually wrote a great article about that book, which I'm going to link to, where you kind of summarize it a bit. And I can also link to the book. But I mean, they, like, as you're saying, they have the model, they have the philosophy, they have the physiology. None of it's exactly perfect or right, but it's setting forth this kind of orientation of trying to think about these subjects in a bigger way. But were you doing that? I mean, were you reading that book, thinking that way?
Oh, yeah, I think, I don't remember.
You don't remember?
I think so. Yeah, not,
Well, if you were with Jim Rank, then I guess that atmosphere was similar, and he had already been there. And yeah.
no, it was, that was part of a basic reference to part of what we were, thinking about and talking about.
And so how did it progress? Because I want to mention place cells were the first, and that was basically, what can we think of it as, like, how do we describe a place cell? A kind of a cell that's representing space in a way. Can we say that in the hippocampus? Yeah. But then
the broad interpretation, it tells the animal where it is. It can be described as self-localization. Okay. We are animal. Yeah. And second to that, what can I do when I'm here that I can't do when I'm over there? Right. What opportunities do I have here that I don't have over there? Or maybe over there are opportunities I want to get to. Right. So that gets to navigation. How do I get from here to there, where the opportunities are different than they are here? And this is where it gets a bit tricky, at least for me, because it's not that the animal needs to stop and think, this is where I am. It's happening immediately. I mean, in a way, it's the body and the environment, and this is happening. There's no gaps where everything stops. It's ongoing all the time. It's the way the animal has, from day one of its birth, been in its environment, matched to, Buzaki might say, its environment. But that's not the only cell, right? So then, when did the head direction cell come? Can you tell me about that? One thing before that, which is, we'll call it remapping. Yeah, that's the thing that really came out of the 3 behavior experiments was not only were there place cells in the 3 environments, but a single neuron could have a place cell in the 8-arm maze, a place cell in the operant chamber. And by the way, these 3 devices were all in the same position in the room, and they were constructed so the animal could see a lot of the room from the operant chamber. The radial arm maze is flat, so you can see the room easily, but the other 2 are, modest walls, so the animal could see lots around it. So they're really physically in the same place in the room, and yet an individual neuron would have place cells in different apparatuses that were really different. The location was never the same within the room frame. And for someone who doesn't understand, are you recording the same cell in all these different spaces? The goal is to and the same cell is showing different firing patterns. One cell might have a firing property in one arm of the radial maze. The same cell might fire adjacent to the bar in the operant chamber, and the same cell might never fire in the home facade. That was one of the things we found, was some cells just were off in 1 or 2 of the apparatuses, and good place cells in the other. That's an important part too. That was the first finding of what eventually came to be remapping. These are our first finding of that. So did that tell you the importance of context and environment? Sort of, yeah. Yeah. Because so the idea that the hippocampus was important for context, as far as I know, dates back to Richard Hirsch, who wrote a paper on hippocampus. There it was just memory and context. And then 8 or so years later, Nadel and Wilner revisited that and tried to interpret it in terms of the hippocampus as a cognitive map. Didn't really go very deep. It didn't get close to remapping, but they did sort of say how this fits with the spatial firing of neurons. And then, so in the pseudo papers that Tim and I wrote, we called it context. Okay, so you were already using that term. Later, I know you used it a lot, but we'll get to it. But I want to hear about the head direction cells, but I also wonder if you had started studying the hippocampus and thinking of, did you go and look at all the stuff of Penfield and HM and Brenda Milner? I mean, were you aware of that whole literature and everything? So a couple of things there. So now, we have there's O'Keefe's idea, or the hippocampus is a spatial system in rats. There's HM in Brenda Milner, and the hippocampus is important for episodic memory. how do these fit together? Is it the same
Yeah.
organ doing two different things? Is the human hippocampus nothing like the rat hippocampus? Is it, or is there a way you can tie these things together? So that's become sort of the holy grail for the field. Can we get these things to merge?
Mm-hmm.
And there's some ideas from the early on about how place and memory overlap and how they may be related. But more recently, they've begun to crystallize a little better. But it's still, those are two very big different things. And the third thing was in the early 70s, long-term potentiation was discovered. So you have HM and learning, episodic memory. And then the ongoing question, well, how does the brain store memories? How can it store, what is information? How is it stored? And there were various theories. And probably the most important discovery was this thing called long-term potentiation, where they were able to show that if you stimulated an input pattern to a neuron in the right way, the synapse could change its strength. And the idea that synapses are not rigid, fixed things, they can strengthen and weaken, and that can be the engine for storing or losing memory, so-called synaptic efficacy.
Yeah, and you had this Donald Hebb.
Right, and Hebb, there's something called the Hebb neuron. Donald Hebb was a psychologist who talked about how simple rules for strengthening or weakening synapses would explain memory in some fashion. And then Lomo and Bliss demonstrated this LTP, which seemed to fit with Hebb's idea.
Yeah, and that idea generalized not quite in the right way, but the way people might recognize it is neurons that fire together, wire together.
Yes.
And this was kind of an actual real experiment that showed something deeper than that in a way, but demonstrating that.
Thank you.
Yeah.
Yes. And so as I wrote that blog post, you were talking about there's nothing about LTP or synaptic, how memories would be stored. Although they talk about HM and learning and stuff like that, they don't. And I asked Lynn, did you consider that? I said, oh yeah, Bliss was down the hall. We knew all about it, but they didn't.
Oh, wow.
It's interesting that it's not in the book, which is a little surprising. Anyway, so these
I guess some edits had to be made.
I don't know. So these are the three big areas of work that centered on the hippocampus. And can you make any consolidation which brings these three things together and get to unified function of how the hippocampus does its job? And
so we know the hippocampus matters for things like memory, and we know that the hippocampus matters for place. We're learning how those might be very much connected. We're going to get to that. But at this time, it was still very new that this area of the brain had anything to do with navigation. Now there's been a Nobel Prize awarded for that. But then this was a new beginning. You were in the middle of it. So how do you remember it kind of growing, becoming, moving at that time? Because place cells was just the beginning. There were a whole family or a zoo, as I think Tim Behrens calls it, of other cells. And what in the Nobel Prize they call a GPS or a sat nav of the brain. How do you remember this time period and the other cells?
I'm going a little chronologically, and you've asked about head direction cells, so now's about the time for head direction cells.
Okay, let's do that.
And that's Jim Ronk.
Who you're working with, who gave you the 3 behavior task.
Right. So he came, as I remember it, and I was beginning to work with Bob Muller. We'll bring him into the story soon.
Another person that's important in this whole story.
And Steve Fox was a very important person. We can talk about that a little bit. So I remember Jim coming over and said, you guys will all work in the hippocampus. I'm going to work in retrohippocampal areas, see what's there. Huh. And so he was just putting electrodes down in subiculum and postsubiculum in different places and seeing if anything interesting popped up. And he got to the postsubiculum, which is ambiguous, and also the dorsal presubiculum, and it's adjacent to the hippocampal formation, also adjacent to entorhinal cortex, and found neurons that had very clear correlates. So the whole job here with the O'Keefe and Ronk were doing was putting electrodes down and just letting the animal behave and saying, Does the cell tell you anything? Does it tell you a story?
Mm-hmm.
And the issue of signal to noise is very important. Because, I mean, you could well imagine the cell go pop, and then it would get to near a piece of food. It might increase its firing rate a little bit, and you say, "Oh, that's a food cell." But that's not what we're looking. If you're looking at this thing one cell at a time, you want a really robust correlate. And the plate cells are very robust correlates.
Mm.
Head direction cells put plate cells to shame. They
Really?
They're astounding.
Can you tell us about those? The title of them tells us a little something,
Yeah.
head direction, and
It's actually
talking
not
about a
that
GPS.
clear,
But
people were somewhat confused by it early on. So again, the same basic idea, the rat's got an electrode in its brain, and in its post-orbiculum instead of the hippocampus, and recording and listening to the firing of single cells. And I said Jim was looking in a few areas, and most of them were sort of confusing or not very obvious. But this area, the post-orbiculum, he found neurons, and the neuron would fire like a compass. That is, one neuron would fire whenever the rat's nose was pointed, say, northeast. Another east. In somewhat random, but each neuron had its preferred compass-like firing direction. Wherever the animal is in a pretty big apparatus, it was still northeast. It was absolutely not place, had nothing to do with it. It was head direction, totally independent of place. And as I said, the signal of choice was through the roof. These cells might fire 80 spikes a second, which is almost as fast as, almost beyond hearing individual pops, within a narrow range of maybe 80 degrees head angle, and then any other direction, silent.
How did he figure out it was the head angle?
Because he just watched it. Oh, he watched it, and then he picked up the animal and pointed its head different directions.
And then,
it was
incredible.
Yeah, I mean,
must have been quite a moment.
The place cells, the animals pretty much have to be locomoting. Head direction cells, no, you can just pick them up and point their nose, and you can lift their head up, and not much difference. But side to side is the important variable. And
do you remember when he found this?
Yeah, I mean,
was it exciting, or was it like, maybe this is something?
Well, he says
you imagine these like eureka moments of everyone running through the halls.
On a Sunday, and no one was in the lab.
Oh.
In his story, he went to a party that night with friends, none of whom were scientists, and he tried to explain it to them, and not
that sounds fun.
Great success. But I do remember very clearly Jim built a very long, maybe 30 or 40-foot recording cable, and our recording rooms were sort of small closet-like room, a little bigger than a closet, with one adjacent to another in a big common lab space. So one question was, what happened if you moved the animal really an appreciable distance from, say, one of these recording rooms to another, one the animal had never been in before, as far as we knew. And as I said, you can just carry them and get the basic phenomenon. And so he built this long cable and found a nice head direction cell in its familiar room, and knew, I forgot the direction reference, but saw that, carried the rat in its cell into, and the rats, by the way, are, they're really friendly. You get to know them very well, and they don't mind being carried, and they'll sniff,
We love the rats. We owe a lot to the rats.
Yes. So in the adjacent room, what I am sure is, our memories are not identical, but I'm right. The cell, this place cell, initially was very quiet. And after a minute or two, began to fire a couple of spikes. And after maybe 2 minutes, became robust again with the same heading direction. So, a question you can ask is, how did the animal maintain its sense of direction going from one room to another? That's an interesting question. Basically, to get that, you have to sort of carry the animal carefully so that it doesn't get disoriented.
I see.
It keeps a sense of direction from one place to another.
Okay.
If you just put it in a box and it would get lost. But by the way, it's not 100% compass direction in an environment like, if you move the wall cues, the field, all the head direction cells will rotate. So it's not as if it's magnetic sense, always northeast in terms of pure northeast. It's
okay. That's important. It's not a compass in the sense of actual,
And that was shown in the first papers, that very beautifully you could rotate the environment the animal's in. If you did a sort of secretly, so the animal is not aware you're doing that, you'll fool the place cells head direction system.
It's very beautiful.
Not compass.
How would you explain that if you put it beside remapping of place cells?
Yeah, so the idea of remapping of place cells fundamentally is, if you have a whole set of place cells and you go to a new environment, they scramble with respect to each other. That's what a remap is. It's not with respect to the outside. It'll effectively be with respect to the outside world, but really the map is the place cells with respect. So if two cells have overlapping fields in one environment, they won't, the chance that they will in the second environment. The head direction cells do not remap. They can change their globally change. They can get fooled, but two head direction cells with, say, neighboring directionality, they'll always have that neighboring directionality. The whole system is coherent wherever the animal is.
Wow. So I just have to pause for a minute and say, this is really amazing, right? It's amazing that this is how the body works. I should also say, for those who don't know, that this was only in rats at the time, but now we do know that the human hippocampus, which is different from a rat hippocampus, but nonetheless does have these. It's been shown with experiments. And so we can say that the human also has this system.
Data comes from bats. Some of the bats data comes from bats. Wonderful stuff coming from bats, and they're flying and how they navigate and stuff like that.
Yeah, so and bats.
Rats aren't necessarily and more mice, but bats are really fantastic.
Bats are fantastic. We could do a whole show just about bats, but let's bracket that one too.
But find somebody else.
But there's something that seems like there's something missing that we haven't talked about, especially when you're talking about the remapping of the place cells. Like, how do they know how to do this? And so what else is in the GPS that might help us understand this system a little bit?
So there are two other cell types that have been looked at carefully. So one gets a lot of attention are the grid cells, discovered by the Moser group in Trondheim, Norway.
And actually, the Mosers and O'Keefe are the ones who won the Nobel Prize. But
right. And so I hope now people understand what place cells are like. So as individual place cell will have one, maybe two spots in an environment where it fires. Grid cells also fire in locations in the environment, but a typical grid cell will fire in many locations. But what's amazing is that these aren't just randomly related to each other. They lay out almost perfectly in a grid-like pattern, hexagonal lattice.
Yeah,
I think you'd have to see the pattern.
I'll show a picture of it because it's so beautiful. The place cell, I'll show it, but as you were describing it, you have the space, and then kind of, if you're going to just draw on where, or make dots, let's say, where the neuron fires, it'll be kind of one blob in that whole space. But then when you look at the grid cells, it's really like a beautiful kind of honeycomb-ish metric, right?
Yeah, it turns out you have to use a pretty big apparatus to see these multiple hot regions, but once you see them, it's just fantastic.
Is it true you reviewed one of the early Moser papers about that?
Not about that.
Okay.
They were trying to record place cells in a water maze.
Oh,
really a challenging task.
Yeah. With Richard Morris. Yeah. Okay. I won't go down that rabbit hole. That's another interesting.
And
so, what do grid cells do in this GPS? Which
we don't really know. I mean, there are a lot of ideas, but these cells all interconnect with each other. The grid cells are not in the hippocampus; they're in an adjacent region, entorhinal cortex, a region that has interconnections both ways with the hippocampal regions. And the head direction cells are in another adjacent region, this post-subiculum, with interconnections principally with the grid cells more than with the place cells. So in another cell type are these called border vector cells, that cells fire with relationship to obstacles, walls, a certain distance from a wall. And so these are like picker toys. We can think about ways that these fit together and either reinforce each other. One early idea was that place cells were downstream from grid cells. If you add a few grid cells together, you get a place cell. Well, it's not that simple, but they do interact. and you play with
Yeah.
one group and it affects the other group. And I don't know that there's any simple answers.
Everyone's still trying to figure it out, but obviously
Right.
there's something beautiful happening here. We have cells firing for borders, cells firing for head direction. We have cells doing some kind of weird matrix of the wherever it is, metric. And then we have the place cell, which is firing in a particular place to generalize it in a way.
Yes.
So when did you start talking about spatial firing? Because I want to move a little bit into your work with Bob Mueller and the stuff that you developed.
Good. Yeah. So Bob Mueller was a little older. He was a faculty member at Downstate working in other stuff. And he had a very interesting background, both in psychophysics. He was really this hardwire, hard science scientist. And he also had a background in psychology, in soft side of psychology, up in Canada, working with Case Vanderbilt, not at McGill, but at institution.
Had he studied with O'Keefe somewhere or something too?
No. Were they friends? They knew each other actually indirectly from college. So Lynn had talked to you about how Lynn Nadell and Bob Mueller were classmates in high school.
Yeah, they were sitting in front of each other,
both in Manhattan,
behind each other or something.
Stuyvesant High School.
Yeah, right. At the
elite high school in New York.
Mm-hmm.
And then Mueller went on to City College. And a couple of years later, O'Keefe went to City College.
Ah, okay. And so Mueller was just a bit ahead of him.
And so they both, I think the guys, they both had this mentor, this guy named Phil Ziegler. I think that's right. And Ziegler,
yeah, I've seen that, Ziegler.
So told O'Keefe, you should be more like this guy Mueller, who was ahead of you. He's really good at this.
Oh, really? Wow.
And so I don't know.
So he did hear his name early on. That's funny.
So it's funny how the New York connection overlaps.
It's funny what a small world it kind of seems like when you look back at it. Everyone's bumping around each other there. And I want to take just a pause because, again, I talk about McGill and UCL all the time for good reason, but downstate too, right? So all of you, it was Ronk, it was you, and Mueller ended up there, right?
Right. So after the fact, we call it the hippocampal group or something like that. It was a wonderful atmosphere. So one of the things I think Jim brought to the place cell group, to O'Keefe, was he really came from physiology. He was he had been studying impedance in the brain, the most boring kind of thing imaginable. How putting, just how resistance to the brain changes when you put the electrode in different positions, and somehow jumped to recording hippocampal neurons and behaving. I've asked him 1,000 times. I sort of understand the jump, but it's not completely clear to me. Partly,
let's pause just a minute here because this is important. I think this is kind of different communities, isn't it? The physiology and this, because you're about to start developing a lot of technology, and sometimes I feel like maybe that's part of it too, that this side of it doesn't get talked about a lot because it's a little bit of a different perspective on things, even though it's part of the whole evolution. I mean, in the kind of more psychological. So what is going on here with the physiology?
So this is my looking back on it. I think, the McGill group, Keith and Nadell, they really, doing great physiological stuff, but were asking psychological type questions. And Jim Ronk came from a physiology department at the University of Washington, physiology at Michigan, then physiology at Downstate, came from a deep physiological orientation. So one of the things he began to do, largely with Steve Fox, was say, okay, what cells in the hippocampus are these place cells? There are lots of people know that a lot of different cell types in the hippocampus. And they pretty much established that they were the pyramidal cells of CA1 and CA3, that the other cells, the whole categories of interneurons, were what they call theta cells. These were cells that increased their firing rate when the animal was walking in the theta rhythm. In the theta state, with the theta cells firing a lot, was critical to getting the place cells behave as place cells. So they, in that sense, began to put the hippocampus into a physiological context.
So for someone who has no idea what physiology means, it's more like, how would you, instead of
the brain, the tissue of the body.
Okay.
Not so much as a psychological organism.
Right.
It's not.
So you're really looking at cells and the actual,
how the neurons, what their membrane potentials are, all the characteristic of the cells of the brain, without paying direct attention to the psychological or higher level functions.
Right. So it's really two ways of approaching it. And strangely, it overlaps in terms of findings, but it can get lost or confused or seem like people are saying different things sometimes without understanding those different approaches, at least in my experience.
I think politically this was sort of important also, because Jim came from physiology.
Right.
And when O'Keefe was claiming these things, the physiologists, I think, feel were saying, oh, what are psychologists doing recording from the brain? They don't know what the hell they're doing.
Yeah, I can imagine if you're coming from a certain perspective, it seems strange.
And I know that, I could pinpoint a few of them. And O'Keefe, had big resistance from major figures at the time. And most of these were physiologists, people doing the hard, the difficult electrophysiology with huge racks of equipment and things like that.
Yeah, I think that's important to even imagine that world. I was going to ask you, like, what does it look like in the lab and stuff? Because it is a real orientation and reference, and it's a real world, right? And if you're not in it, it's, I think, hard to imagine how all-consuming it can be, the way you can kind of see the world through it. I don't know.
Yeah. I remember myself going into, say, Jim's lab, which wasn't this hyper fancy lab, but there were racks of recording equipment. And I said, my gosh, I have to learn what each of these things does. And,
But I thought that's what you wanted. That's why you went there. You are
right,
also, yeah.
And this was a fairly simpler set. There's some systems with 4 or 5 racks of stacks of equipment, and the idea that you could just understand what the signal processing. This is before computers were playing. And computers
exactly. You had to really understand it.
Right. Computers were just beginning to play a role in neurophysiology. So some of these labs were on the forefront of bringing computers into the labs. O'Keefe was not, for example, and the McGill group was not, but the physiologists were. And big computer machines, pretty expensive stuff by research standards. So it was a different
that says a lot about the different mindset already, right there.
Right. So Jim,
computing
with his very open style, and having also being a little older than O'Keefe and Nadell, and being from the physiology community, I think helped warm some things up and help make entries into the physiology world. I do remember when I was a postdoc, we were beginning to record plate cells. Visiting neuroscientists would come by, physiologists, and they'd come and they'd watch me recording a plate cell, and they'd say, "Oh my gosh, that was," they flipped. They would see what we were talking about.
Yeah, because you can't imagine it until you've seen it, I think. At least not back then. Could you compare it to anything today that's
so one big impact was functional imaging.
Okay.
And that started roughly in the early 1990s. And so as I alluded to earlier, cognitive neuroscience is really pretty new. And it's expanded a lot in the time I've observed the fields. Yeah. And one of the things that made it expand was O'Keefe and the Mosers and recording plate cells in the middle of the brain, where you shouldn't go if you want to figure things out, and yet something popped out. The classical neurophysiologist said, "No, you're crazy to put electrodes there. Work from the periphery and figure it step by step." But lo and behold, interesting stuff came out.
Yeah, that's an interesting point too. That was also the prevailing sort of philosophy of the time, right? Start at the edge. Don't go in the middle.
Right. So actually, the Mosers say that very nicely in their Nobel Prize speech.
Oh, do they? I have read that, but that's probably where I got it from.
But and we felt it as well. That the famous studies were the visual system going from the retina to the lateral genicular to visual cortex, and that's how you should proceed, and watch how the signal gets changed and extracted. And for the motor system, the inverse of that. But putting the electrode in the middle of the brain and seeing anything and interpret, people would say, well, it's like recording from a single transistor in a computer. You're not going to learn anything. It's not going to tell you anything. But maybe it did, and people were lucky, and it did.
I think it definitely has.
So it wasn't guaranteed to work.
That's kind of the beauty of this too. It was a little bit of an accident in a way.
Yeah, that's the current fight, because people still say single cells are the wrong way to think about it. You have to think in terms of networks, recording tens of thousands of cells, and don't even get a hint of an idea from single cells. So the argument is ongoing. Yeah,
and I would say they're both good arguments. I mean, they're both important. But I want to talk a little bit about your work with Bob Mueller. So what did you guys start doing? You're spatial firing, you're developing new technologies.
So as Mueller came in, wanted to work with us. And he knew about the place cell stuff. He knew about O'Keefe and Nadel. He somewhat friendly with O'Keefe and Nadel and all that kind of thing. And so he just said to me, let's collaborate. And the rough idea, and I can't even pinpoint how the strategies worked out. It was between Jim Ronk and Mueller and I and me, between us, among us, whatever the grammar is. Anyway, was to simplify, definitely to have some camera-based recording system for where tracking the animal in a very simple apparatus. And the apparatus was a cylindrical enclosure with very simple sensory cues. We put a white cue, a big white piece of cardboard along one wall, about 90 degrees of the wall. Oh. And the animal's tail, we just wanted to cover the apparatus without interfering with the animal behavior much, without going in and poking the animal. So we trained the rats to expect food pellets to drop randomly on the floor. Initially, I was throwing them over the curtain and walking around the curtain. It was curtains, by the way. Finally, we had curtains enclosing things. But later, we had an automatic dispenser that was scattering pellets. Oh. But so the rat pretty happy walking around, and it will eat for 15 or 20 minutes before it wants to drink. So that was roughly the length of the sessions. And the camera was. And so it required a computer. And this was.
Is this the first entry of the computer into this process, other than a video?
In terms of, in parallel, McNaughton and O'Keefe were computerizing also, almost exactly in parallel.
Yeah, that's also very interesting. But from your perspective, what was happening? So why did you want to do this? What advantage was it going to give you?
Oh, lots of advantages. One was O'Keefe's description wasn't overly convincing. Mm-hmm. People thought it's subjective. He's telling you where it's, he's just making these little circles on the floor. So
and to this idea that you were saying, one cell and so on. I mean,
so we wanted to take ourselves out of the, system of collecting data and demonstrating the data. But also we could, it's a much more refined, potentially more refined way of describing what's going on.
So in doing this, could you change the environments?
I mean, so initially, no. I mean, with 2 papers, we came up, initially was just documenting that they're plate cells. And
so you were able to get a more robust documentation that this is actually really happening.
So we did, I think, many of the appropriate controls. And so 60 times a second, the animal's position is detected and how many spikes occurred in the target cell. And then collecting this in a chain over 15 or so minutes, and then putting it together and taking each pixel, each location, and saying, okay, how much time the animal collectively spend there, and how many spikes were there, and compute a firing rate for each pixel in the apparatus, and then making a map of the firing rates. And we use the color coding system, which was very expensive to publish. So we had these nice color pictures of these bullseyes of place cells on a yellow background were pixels that with this rat visited plenty, but there were no spikes. And it was, these were pretty pictures that were pretty convincing. And so we did this for a number of cells, and we could characterize the size of the firing field, shapes a little bit, and the distribution, that kind of thing, within a single apparatus. And the other aspect was then begin to change the environment on the animal. And before
you go to the environment, can I ask a question? It's kind of asking you to be a little egotistical, or not really. But do you think this played a role in solidifying the people we're going to accept? Yeah, place cells are real, or this.
I hope so.
Yeah,
I think so.
Because
we weren't alone, as I said.
A lot of people say that this work with Mueller and this is important, but I'm not sure that how to make it really obvious. But I think one way, obvious to people who haven't read all the papers, but one way is that it did show that it's legitimate. Is that fair?
Yeah, I think so. Yeah. And so we summarize the second study, which was going to different environments. Mm-hmm. We really demonstrated the remapping. We didn't use the word remapping yet, but we showed the remapping, that the cells individual or pairs of cells would go to independent locations, and some would turn off in some environments. And the environments we used were cylinders and boxes, and double-sized boxes and double-sized cylinders, which was real interesting because some of the cells in the double-sized one would just expand. They would show some, and others would just remap or change. So that was a partial effect. But the expansion was very interesting, and told us something about the nature of what's going on here. And
do
then
you
we
remember what your feelings were at that time? Was it exciting, or was it?
Oh, it's exciting.
Were you trying to piece together a larger puzzle, or was it more exciting from the technological advances?
Well, it took us years to collect all the cells, so
yeah,
it wasn't
it's such a long process.
It wasn't an aha kind of moment. It wasn't eureka. And I can't remember the stages. But one of the, reading back on those papers, and we knew where at the time, but also reading back, we had some insights which we carried forward. One manipulation we did was to put barriers, little, in the environment. And we found that if you put a barrier in what had previously been a place field, it would disappear. But it wouldn't affect the rest of the apparatus. You put the barrier somewhere else and it wouldn't have an effect.
Sorry. So it would, okay. So that's another environment remapping.
And so our conclusion, or tentative conclusion, was that it was the barrierness. The barrier could also be transparent. It was whether the animal could walk through it or not. That it was the behavioral space that was available that we thought was, a lot of the importance of what was coming at. That in a more common term, maybe wouldn't say the affordances that the environment presented. What behaviors are possible here? What paths I can take? What paths I can't take? Can't go through that wall. I can't go through that barrier. That kind of thing. So we didn't know the word affordances, but phrasing that sort of said that kind of thing.
This is interesting. Maybe we can start to go into some kind of, I don't know, just like explore some territory a bit here and some bigger ideas a little bit, because this makes me think about, so this is very behavior oriented, or the biological process related to how the animal is moving through a space. And it makes sense that if it can't continue forward, that the physiology would change. I mean, it just kind of seems to make sense. But let me ask just a big question. Do you think memory is that kind of behavior too? If we think about Tolman and the cognitive map and the idea of just the rat, let's not think about our understanding of memory and thought yet, but just the rat in the maze, and the idea that it has a kind of cognitive map, which is a kind of knowledge, which is a kind of memory, or is it not? I mean, it's not episodic memory. It is. I'm trying to get to, if you see any connection here.
Yeah, so my rough notion, definitely memory, but not memory in a simple episodic sense. There are sort of two layers. One layer would be the substrate or the background upon which episodes can take place. So if you have an environment, say an environment covered with place cells, it permits certain paths to take place, with the paths being roughly equivalent to the episodes, but the context or the environment are upon which the episodes rest. You could not have these episodes without a background. And that's sort of a fundamental, I think that's in contrast with Eichenbaum's rough idea that it's all associations. And this is what might be called a two-layer approach, a background layer, and then a episodic layer.
Okay, let's try to unpack that a little, because for some reason I'm thinking of Goff. I mean, now that's kind of going to humans, because I think you wrote something very interesting about golf courses. There was a article in the New York Times way back about how people who play golf have better memory, but it's subject to better memory of the golfing experience, if I remember correctly. And I believe you said, I think the article said it was for one reason, which I don't know what, maybe it was just that people who play golf have better memories because of the action, but you were kind of relating it to this method of loci, of the space itself being important.
I only vaguely remember the golf
Oh,
story.
okay. We can
But
skip that. Yeah.
but I agree with the rough idea that
Okay.
if you have, a very clear substrate that's really rock solid,
Uh-huh.
that memories within that space are easy. I think of, people who can play bridge and recall a bridge hand.
Yeah.
Everything. I've tried a little bit. I can remember a couple of them, but people who really do a lot of, or chess game, they can reconstruct the chess game entirely. Part of it is by chunking and things, but the context of the experience in chess game, I guess it's the chessboard and the affordances of all the pieces or something like that.
Yeah.
And I think that once, if you have a really rock solid background, then the stuff that lays upon that is much easier to extract. I heard a similar thing about wine, this wine specialist who could identify the field of origin of grapes of wines he was tasting. And it sounded impossible. That globally he could tell you was some field in Australia or something like that. And he was interviewing lovely sounding guys. It's not as hard as that. First I said, it's really not that hard. First I say, oh, this has got to be an Australian one. There's no question about that. And then from there, I can narrow it down. And so it's a two-layered approach. And I think that many of these things have this solid background foundation upon which a specialist memory can be laid.
How much does that have to do with experience? I mean, if we're talking about the rat that's born, if we think about its whole development, it encounters a really certain kind of, well, does it? Let me ask. If these rats are born, are they born into the situation of the experiment such that basically they only know these kinds of environments so that,
yes, both two major lab groups, the O'Keeffe's and the Mosers, have looked into this, mostly the Mosers, I think, of trying to look at, place cells, head direction cells, grid cells, which come first, what experiences are critical for one emerging or the other. And it's not that clear a story. These are technically very challenging things. And so one of the things is technically to record from the brains of these very small animals. And the second thing is to manipulate the environment in interesting ways so that they don't have, say, these rectangular boxes that they are growing up in. It's somewhat different. And just not all that clear. I think the rough thing that I would remember from them, apologize to them if I'm wrong, is that roughly head direction cells seem to roughly precede the other two categories. And second,
it's kind of like a tuning mechanism then or something.
Yeah, I think that there's some, people talk about hardwired and not, somehow the whole notion I find a little wrong.
Hardwired is a rough term.
Yeah, I mean, because everything's dependent on some experiential state.
And we're all developed in different ways.
But
here's what I'm trying to get at, though. How important, how fundamental is place to memory? And how fundamental are the patterns that you develop in place? I think like if you put that barrier up, needing to find a way around. I don't know.
My bias is that the place, so there are two rough ideas. One is that place is not fundamental, it's just easy to see. That you could have any low-dimensional framework equally good.
Is that more Eichenbaum, or it's a matter of relation? It doesn't.
No, Eichenbaum wouldn't even be that. He would say there's no low dimensionality is not really relevant.
Okay.
But it's something like that. There's no background set and foreground set. But these people would say there is a background set, but space is not really special. It's just easy to record. And O'Keeffe and us, we were lucky to be able to see that so readily. I think that's not true in two ways. One is place jumps out all the time, these place cells. You want to see it or not. You can put the animals in tasks that are not very spatial, and the space things jump out anyway. The second thing is, I think, there's not much data here, but I think from an evolutionary perspective, and my guess, this is mostly guesswork, is that the hippocampal system in snakes, or reptiles, back to snakes, was fundamentally a navigational system. It didn't do these other things. Didn't have to do with episodic memory. And when increasing demands were put on the system, could you help us with this task, with that task? Then the spatial framework got reused in different ways. And so this is a complicated story to put forward, what the hippocampus is doing in mammals and humans, how it relates to memory and episodic memory, and how it relates to navigation. There's no proof or simple stories here, but I do think that place and spatial location-specific firing at any rate, what can I do in this spot, and something about navigation are substrate, they're foundational, and then other things build upon that.
Okay, so I guess what I'm trying to get you to say, I don't know if you even think it, is that place is fundamental to memory. And then I wonder, is memory also fundamental to place? Like, I feel like in a way, from our position as humans, these seem different.
I think it is.
Are they?
So, jumping back in evolutionary time, think of some reptilian or like ancestor who comes out into the world. Now, the world is not preformed. There's no way that the features of the good places and bad places would be hardwired into the
right. So the creature is not coming into the place with the little map in its head of the place, because this doesn't work. Yeah.
So to make use of this machinery that knows about places, it has to learn about places. And so that, that's as much of an argument as I can make it. It's not, all the snakes or whatever aren't born into the same area, in which case they could have inherited some map. That just is not realistic.
But they are born and they immediately must navigate and make way in that environment. So
methods
they're forming
of navigation.
those.
Yes, navigation is not all map-like or cognitive map-like. There are various effective ways to navigate, and I'm not even sure the degree which humans use maps. I mean, many humans are not very good with maps, and having an aerial view of where you are doesn't always help you get from here to there.
Right, and I think sometimes we confuse map and territory, that even when we talk about a cognitive map, it doesn't mean we're pulling out a little map inside our brain. I mean, this is the homunculus argument. So it does get kind of confusing, and we won't get into the weeds there, but I wonder if you agree with this. Like, if that snake or what that's creature, amphibian, is born into its environment and it immediately has to navigate it, make its way through it. And this is obviously creating some kind of alignment with the body and the space in the same way that the rats learn, or if we think of Tolman, the route to the reward. That's learning, I guess, right? That's learning. Does that then set forth a kind of pattern?
Well, there are various ways you can optimize navigation. And I think one not easy way, technique, it's not so easy to understand, but really used by animals, is path integration.
Yeah.
And so this has been beautifully studied in desert ants.
Oh, really?
Yeah. So
I need to write that down.
Weiner I'll send you the name of the principal guy or the papers. So imagine a desert ant has a home base, and it's wandering out to look for food. And it takes a zigzag path, foraging through its environment. Now it finds food. These ants can go meters and meters across sand, no information from the substrate. And after they find food, they can take a direct path back to home.
Yeah, amazing.
Now, that's not a cognitive map. And what's been shown, what they effectively do, is for each leg of the outbound journey, think of each leg as a vector, a straight line from one spot to another. Add up all of those x vectors. If it were Cartesian in x, y coordinates, you just add up the x's and add up the y's. Okay, so you have an outbound vector, and then you just invert it to make it all negative, and you have a vector that points directly back to where you started. Mathematically, it's beautiful. How the world the ants do it is astounding. So what the ant has to do is keep track of two things: how far it goes on each vector and each leg, has to count its steps. That's called odometry. And it's got to keep track of its head direction. And so each vector is a mix of distance and direction. It keeps them, you can think of it as XY coordinates, but they almost certainly are not doing that. That's the way we can think of it. So those are the vectors.
That's the
Add them
way
all
we can
together,
model it.
invert it, and you go right back. And the ants are incredible. It's been shown that they have this directional sensitivity and that they count steps. The counting step method in ants was to make stilts on their legs and show that they miscounted, or
Oh,
to
yeah.
cut little stumps off and they miscount the other direction. It's a really beautiful work. But that is a rough summary of one kind of path integration, which is how you get back home. But path integration can also work connecting to locations within a familiar environment. And so path integration is one that's not white map-like. It's got features of distance and direction, which are real spatial features, but that's one method animals can. And I think we have to assume that animals exploit all kinds of functional methods for navigation.
Yeah, it's not either or.
And so
or only one
the
even.
gold standard, like the water maze standard is, can an animal take a novel route to an unmarked goal? So in the water maze experiments, the animals explore the water. I mean, they have multiple trials where they swim around blindly and find the platform, which is a way to get out of the water. And after multiple trials, you can introduce them from a new start location and they can take a direct path to the platform, which gets them out of the water. So that has, as you pointed out earlier, it's become sort of the robust standard technique to demonstrate some high-level navigational ability. Exactly the strategies the animals are using, whether it's really map-like or path integration, whatever, is a little unclear to me. But it is an efficient and effective strategy, and one that's pretty easy to reproduce. It's very difficult to record place cells in those environments which the most was discovered. So it's not a great environment for physiology. It's very good for behavior.
Well, let me just put something out there that maybe you're going to totally disagree with, but I'll use it to see how you think about this. What if the ants, of course, they come into the world with certain bodies and they're aligning to their environment. It's a certain pattern. So in a way, the dead reckoning or the path integration, the body itself is providing something like what head direction is providing.
Oh, yeah, it's now pretty well worked out in some insects what the directional system is. That they have a head direction system. It's with respect to the polarizing cue of the sun, but they have a directional. The neuronal mechanism is very similar to head direction.
But it's not that. I guess what I'm saying is we don't have to necessarily think of it as they have a GPS. They're not like pulling out a little map in their brain.
No.
But they are the GPS. They are the GPS. They are the dead reckoning system. They are the path integration. I mean, could this be just happening like you said, affordance, almost in an evolved Gibsonian way? I don't mean vision.
For the insects, I don't know that they had to learn anything. They do have to keep a running tally of the vectors. That's all they need.
But that could just be happening with the body.
Right. I mean, but we talked about consciousness. There's no reason I would say that they have to be conscious of what's going on.
But couldn't that be, I mean, I guess what I'm saying is if we think of that's just happening, it's hard for us not to imagine they're having a little map in their head and they're aware of themselves. But actually, they're just, as we do, as babies do, you come into the environment, you must navigate, you must find way. Your body itself becomes your GPS system, not just the brain that you're pulling out a little map. But could that be continuous in the sense like when we learn something like language, and probably other animals have languages, but I'll talk about human language, that then becomes a way that we've communicated with the world and come back to ourselves. So that same kind of alignment or navigation through something like language, we might use in a very continuous way that to plan or to do something similar, to navigate spaces that aren't necessarily geographical. I know that's kind of a big thing, but it feels to me continuous if we think about.
I'm not really sure what you're getting. I think I have a little sense of what you mean by continuous. Do you mean
it's the same kind of process?
So
it's navigating.
I'm thinking continuous is distinct from a deliberation.
No, I don't mean it like that. I just mean
so walking is sort of a continuous process. We don't have to pay a lot of attention once we start walking, we can have a conversation with a friend and that kind of thing, and people know what to do.
Yeah.
Is that the kind of thing you're thinking of?
Well, no, but that's actually very interesting. That I would think more of like a rhythm. I guess what I mean by continuous, I mean in a really everyday sense, not in a neuroscience sense. I mean just that whatever this body is doing, like the ant, the rat, or us, when it is born and must navigate, make way through its environment, there's not like some separate thing that then becomes planning and all the stuff we associate with language and image. For me, it seems like you're constantly kind of learning this stuff, and that, so for example, like the little rat, right? When it's in the maze and you put one of those barriers in front of it, it seems natural to me that it would use its same patterns of navigation to, I mean, in a cartoonish way, stop and play out those scenarios that it's already been in, because the body would have developed a way that it can continue to replay those events without it needing to be some completely new thing that suddenly evolved. You know, for me, it feels like part of the same process.
So I'm going to circle back to a word you didn't like, deliberation.
Okay.
But
no, I do like that word, but
okay. So I mean, in this context, you didn't seem to like it. So in mammalian, in invertebrate brains, in mammalian brains, there are sort of two memory systems. So one is this episodic memory system, and let's just say it's involved with deliberation. Put it aside for a moment. And the other memory system we can think of as basal ganglia,
so
where
more
it's
motor
involved with learning skills.
Yeah.
And learning sort of fairly automatic responses to stimuli. And these automatic responses can be generalized. So they could be elicited in a somewhat novel situation where your old strategies might just work. And they're, you know, it's a little bit like Kahneman's thinking fast and thinking slow.
Yeah, yeah.
I wouldn't have said basal ganglia, I think, but that's how I think of it. It's fairly spontaneous things that come out without effort, you know, the least resistant kind of response in situation X. And if it matches one of your well-known responses, maybe overlearn, then just go with it, that kind of thing. And
is this like motor, like we need the motor cortex to learn, but once we've learned the action, we don't necessarily need the motor cortex anymore. It's like a pattern generator in the body or the spine or whatever.
Yeah, that's certainly true in rats, and I'm not 100% sure how true that is in primates and humans, but yeah, that's a little bit like that, where the basal ganglia can, you know, without a lot of thought, if you want to put it that way, generate perhaps a useful response. And the more overtraining
deliberation is a good word. Without deliberation,
these can be very smooth and very efficient. They can be better than deliberative choices. They're faster, you know, if you really learn the response well. You think of a trained athlete, a tennis player or something, they're not thinking about all the components to the stroke. It's all just, you know.
But they did at some point. That's kind of the thing.
They did, right. So I'm not sure that's what you're getting at, but I mean, it's one term that people use.
Yeah.
Is flow. Is flow something that?
Flow?
Yeah.
I think that's all connected, but let me ground it back with the memory and the navigation kind of thing that we talked about, where those seem different, and reconciling them is kind of, you know, what everyone's doing now, but can't quite see clearly. I guess I'm trying to relate it to that in a way. If we think about knowledge, our conscious experience of our own experience, like I don't mean being conscious or aware, but I mean deliberative, becoming able to think of ourselves as a subject and consider our actions or something like this. All of that to me feels like the same kind of process that we've been uncovering with something like navigation. It doesn't feel like those are in a Descartesian or dualistic way, like separate things. And I know that's getting a little philosophical, but I wonder how you see it.
Yeah, I think, you know, what you're saying. So the experiment I'm thinking of that's closest to these lines is from Pfeiffer and Foster. Are you familiar with that?
I think so. Is that the one where the rats are? Is it a rat one?
Yeah, rat one.
Yeah, and where they are kind of deliberate. You kind
That's the idea.
Yeah.
So I'll try to simplify it, the description. The animal, a signal comes to the animals. Now you've got to find the buried food. And it's in a somewhat unusual location. And where the animal is at that moment in time is unpredictable to the animal. So the animal hears the signal that now he has to find the buried food. It has to find the heat, yeah. What the animal does is it rears, doesn't move.
It rears, so it's kind of stopping and
Right.
on its two
It-
hind legs.
It exhibits these things called sharp waves, which are fairly slow potentials. And riding on the sharp wave is a very rapid sequence of action potentials. And
gets
this
to
sequence
Bizaki's work,
of
I
action
think.
potentials seems to be imagined paths the animal could take from that location. So now,
Wow.
at least according to this rough notion, the animal rears, one sharp wave occurs that imagines one path, another occurs, it's another path,
Mm-hmm.
a third occurs, that's a third path, and then the animal says, that second path takes me to where I want to be, the goal, and it executes something like that second path. So that fits really nicely if it's really true. There's some issues with how pure the experiment is. If it's really true, it fits into what we think about deliberation.
And it's
if-
not the only experiment that shows
No,
something like that. So, I
yeah,
mean,
right.
there's been quite a few in different ways, but yeah, and it's very exciting. I find that very exciting. And
the other fit into
Yeah.
memory is consolidation during sleep.
oh yeah, so it's something similar than this
Yeah,
replay.
simply. Again, we have these sharp waves. And it's well known, this is uncontroversial.
Mhm.
Sleep consolidates memories. These are just behavioral studies from various sorts. That if you learn something during the day, sleep on it, it's better, it's remembered better, even better than right after the learning episode. Sleep on it. So the notion is that the memory, the episode replays during sleep. Actually, the notion is it's selective consolidation. The important things replay, the unimportant things fade.
Yeah,
There are a
yeah.
lot of experiences the animal would have had before going to sleep. So,
And that probably has something to do with the affordances, like that it wants to remember. I mean, I think of the bees for some reason and the dance.
well, there's a paper that came out just this week
Oh.
that the ones that replay the most are the trajectories that are associated with rewards.
Oh, really? You have to send me that paper. I don't know that.
It's out of Bizaki's group.
Oh, it is? Okay. Wonderful.
Yeah.
Then I will find it.
And so it's not just everything that happened gets consolidated, but there's a selection. They both are replayed while the animal's awake during the task and then during sleep, if I remember correctly. So that's a second thing that brings the play cell phenomena into the memory world.
This is very exciting. And I want to, for some reason, bring up the idea of planning and prediction. And we were talking about this GPS of the brain. And I think when I was talking to Lynn in the talk I haven't actually put on yet because we've had a few talks, but we talk a little bit about what they were noticing in this system. And I wonder if it's what you were noticing too, was there's a kind of planning or a kind of I guess what people would think of now as predictive processing or active inference or all these words that are very, hot at the moment. I just wondered, did you see something like that too?
I don't think so. I mean, I'm using most of the other people's experiments, but I like them a lot. Let me just go a little further. So deliberation. So there's something going back at least 100 years, which is something roughly called the two-stage model of decision-making.
Okay.
And it's roughly what we just described, but it's not in rats. It's about how it's Kahneman's thinking slow, but the idea really dates back at least to William James and others, which is that when you have a difficult problem, you imagine various solutions, various strategies you could do, and you select one to act on. So stage one is playing out various what-if scenarios, and stage two is choosing one for action.
And isn't that like taking, like after you've taken certain paths, you start to the body starts to, the GPS starts to understand certain patterns, doesn't it? So at some point, maybe you can begin to deliberate about those patterns
Right.
based on all this experience.
Yes. So in generating these imagined paths, you have to have a lot of familiarity and some creativity. And the paths need not be ones you've ever taken. And the ability to construct paths across space, taking routes you haven't explored before, would take a system that really understands the layout of the environment, or a two-dimensional or three-dimensional space that's not purely spatial. It could be a social network, but it probably has to be what's called low dimensionality, something like space, to be able to construct routes, possible routes across this space. Yeah, I'm very enamored of that sort of two-stage model. I think the philosopher Popper, one of the famous philosophers, really came across it late in life, and suddenly came to the idea of that determinism isn't real because this is how we make decisions.
Because
we played out these scenarios. I don't know quite how it defeats determinism, but it undermines it. It's not just SR responses.
Well, here's how I think it does, is if we can reflect on our own patterns, that means, I think we only do it together. I mean, I don't think any creature that was just isolated would ever do this, but I do think through this process of navigating the world, and I'm going to get a little metaphorical, we do learn how to reflect, deliberate on all these paths we've taken before, and then we can, as you were saying, kind of choose new ones. That doesn't mean that we can create paths completely beyond our comprehension, but we can, just the same way we can path integrate or do all these other things, put things together in a new way or something. So for me, it can show that there's at least some space of free will, or that there is some purpose, maybe even to becoming aware of ourselves.
I can see the arguments against, well, those paths that are presented, they're determined. You can get into knots about that. But one thing I think that people, which is cool about imagining the future, is, our behavior is not totally determined by our past. We can, in fact, imagine the future with some accuracy. Machines don't do that. The deterministic framework is it's all the path determining the next step. But the fact that our brains can make reasonable, realistic guesses about possible futures really takes it out of the loop. And I'm not sure that gets to that. I don't want to really get into the determinism issue, but I think it's very important that we can imagine the future, that our sense of ourselves has a past and a future. And those are, really critical aspects of sense of self, all these sorts of things we haven't talked about.
Yeah, I don't want to go down that rabbit hole either, especially because it would take forever. But I have to just say too, I do think it has to do with the paths that we open up for each other, and that there is definitely a way in which there's some degree of freedom in that. But that would be a whole other discussion.
Let me just comment.
Yeah.
So the route example I gave you about imagining paths, that's not a socially constructed set of paths. It's the animal doing it itself. So I don't really agree, and that's from what I can gather, that all these imagined paths have to be collectively determined, although that certainly.
But I'm not sure if, okay, there's different degrees of it, I guess. So I guess I mean on a kind of human, where we are now level, then in terms of like the future of where we're going to go, for me, it would be a matter of combined, or sort of not group thinking, but the communication offers a lot more possible affordance for something like changing the trajectory we're currently on. The rat, I think you've set those paths, and it's either going to take one or the other. And maybe I do think that's a kind of free will that it has, in a sense, that it can decide which one to take. I guess that's one sort of scale. And on a wider scale, I would say, yeah, it's going to
I would just make a distinction between deliberation and collective deliberation.
Okay. How do you see that?
Well, collective is when you talk to your friend and you share ideas and you get a better set of alternatives than you would have on your own.
So, here's a kind of a leap, but thinking about memory and navigation and sense of self, identity. I mean, memory is a kind of our identity and our sense of self and so on. But for humans, we think of that as being conscious. I mean, the rat is conscious, obviously, but it's not conscious of itself. Maybe it is. I don't know. But so there's all these kind of levels of becoming aware and all this reflection and deliberation. And I just wonder what's your kind of take on that? I mean, do you see a continuity between, for example, that rat deliberating about which path to take and what now we're also as humans obsessed with, this idea of consciousness and
I see continuity. Yeah.
You do?
I mean,
Yeah.
I could say, okay, I've got to get to the cafeteria. What's the best path to take? I could take the elevator, take this. That deliberation of the sort. I could just head out and go, or I could think about the routes and what I could do along the way. So that's, not much different than the rats. And then let's expand it all very broadly. So I have career goals. How do I get? I have career goals. Not anymore.
Yeah.
Get there. So I have an idea about my life and what its projects are, and I want to get to some endpoint. And there are deliberative choices. I put off some immediate gratification, maybe to get to a future goal standard. I think it's, I don't see any discontinuities. I think it's a continuum from how to get to the cafeteria to how to get to some family goals, how to contribute to society, how to do things that make, that fit my sense of self, my self-worth, those sorts of things. I think that there's no real, I don't see a discontinuity. But I don't think that animals have the same extended, I think there's a whole range of stop points for different animals.
Well, they have different bodies and they're in different contexts.
They know they're going to, those are all interesting issues. The elephant probably do, and those sorts of things.
Yeah, I mean, yeah, I won't go off on my feelings about that, but I do. Let's go back to the hippocampus for a second, because so the hippocampus what is it doing with all this stuff? I mean, it's sending it to neocortex, right? Or so.
Good.
Can we relate that to what we're talking about? I mean, can we just
try
to bridge this?
Jump. Let me see if I can do it quickly.
Okay.
Here's another theory of the hippocampus, which I like, which is that it's an index.
Oh yeah, the index.
And so let's take something like an episodic memory. I, have a memory of what it was like having breakfast in my kitchen when I was a kid. There's a lot of stuff in it. There are colors and people and, the shape of the room and the smell and all these sorts of things.
And for someone who doesn't know what an episodic memory is, we can think of it
like
a movie or something.
Episode, sort of movie-like thing. I can sort of, envision what it was like.
Which is different from, like, for example, another kind of memory, like associative memory, or riding a bike or something. Right.
No, it's it's like a flashback in a movie, that kind of thing.
Yeah.
It's got an episode, like an episode in a book. An episode. So now I don't think all those things are stored in the hippocampus. But the hippocampus, I don't have that memory. So what's going on? So the rough notion is the hippocampus has a set of pointers which project out to the neocortex. And the pointers, the memory is fleshed out by where the pointers point in the neocortex. So it's not the hippocampus. The hippocampus stores the connectivity, what goes together, but the content, the richness is neocortex. And probably the essential things that make it conscious is probably neocortex and thalamus interacting, not the hippocampus. So
there's tons of papers on what neocortex does, and but it is kind of the newest part of the brain that does. Most people do see that as associated with this human experience of something like thinking and thought and so on.
The only thing I'd add to the index theory is usually, what's the fellow's name? I can't come up. Back to, the name's not coming to me, but it's sort of a static index. It's like a library card catalog that tells you where to go to find the stuff. One thing I'd add to that, it has a time-ordered property, which we think of these episodes as having a movie-like or temporal sequence. And I think what this stuff coming out of the hippocampus is, like the sequence of play cell firing, there's a time order that comes out. So the hippocampus.
Yeah, it almost can seem like a
conducts-
script or something, can't it?
Right. Or
Even these cognitive maps, they're not static. They're almost more like scripts that are constantly sort of going.
Analogy I can move. It's like a conductor in music.
Yeah, okay.
That
Music
the
is good.
music and the conductor, is the hippocampus, and then it comes out. The orchestra is in the neocortex.
Right. So you have this environment that has this kind of statistical regularity that we could call music, let's just say, and the conductor is sort of turning that into something that will become like an episode, what we experience as an episode. Could we say that? I mean, we're generalizing, exploring.
That's condensed, a lot of ideas into a couple of minutes, but wasn't sure we could do that.
So then we could almost say that the function of our own awareness, of our own memory, thought, our experience of what we call thought, which is usually language or image related, that could be functional in the same way that the hippocampus is functional for navigating a geographical environment. We might be navigating other statistical regularities like conversations or books or linguistic conceptual spaces in a similar way. Could we say that?
So, getting back to this two-stage deliberation, some of the trajectories you reject might be very dangerous. And you reject them because I may fall off that cliff or something like that.
Yeah.
So that's got implicit functionality in that, survival value, if you will.
Do you ever think about patterns? I mean, do you think more about parts like the neurons, or do you ever think in bigger patterns? Like I was thinking about the golf course and how it's such a pattern that no matter what golf course you go, there are certain regularities, and how that makes it easier to remember. Or music, when we hear certain kind of music, that seems to stimulate or enhance memory. I think you've written about that. So that's kind of, but do you see some connection between this pattern or recognition or regularity of some kind? And
I think that's one of the big problems. I mean, issues is, so roughly we had this remapping idea, which separates one set of memories from another one. But how do you, if you want to fuse two maps, if that's necessary for sort of creative thinking, how can that work?
yeah,
Or, it's actually easier to construct a system which makes lots of totally separate learning systems that don't overlap. But how do you take advantage of them being separate, but sometimes they can learn from each other? And that I have no real answer to or idea about. I think that's a big challenge.
well, let's wind it down because we've been talking a long time. I guess, you're still at downstate. That's what I want to get to. So tell me, like, why didn't you leave? Or has it been as stimulating through all these years?
Yeah, the hippocampal group was just wonderful. Jim Ronk is retired and is somewhat debilitated. Bob Muller unfortunately died. Others, Andre Fenton was a graduate student going through here, then a young faculty member, but he's left. So the group that is not very functional right now. I also do a lot of teaching, and that's been at least half my life, and now it's more than half of my life. I enjoy it a lot. It informs my thinking also. I think it makes me more of a generalist to try to teach, more about neuroscience than just hippocampus and place cells. So I enjoy that. I enjoy a lot of working with the students and the other faculty. I wish, I certainly would like it if the hippocampal group reconstituted more. And I do collaborate with Andre Fenton at NYU. I have collaborations with people outside of downstate, but unfortunately not that much is going on here.
Well, you and Fenton did a lot together too. Last question, I guess, is, well, actually, I was wondering, how did you get into kind of philosophy ideas? Mostly through your daughter, or do you still think about that? Is that part of your life?
Yeah, it's a pretty big part of it. I think it was about 15 years ago. I wanted to teach. I got graduate students together and I said, well, we're neuroscientists. A big topic is consciousness. Shouldn't we learn something about consciousness and have some idea? And at that point, Christoph Koch's book, what's the name of his book again?
I don't know. I can't remember. Conscious. I'll find it later. But
anyway,
yeah, Christoph Koch's book. Mm-hmm.
So it was the, then most promising current book on, and we read it.
It was back when nobody was allowed to talk about consciousness, I think. And
right.
kind of shattered that.
And started out with a definition of consciousness from John Searle, which is consciousness is what you have in the morning when you wake up and you lose at night when you go to sleep. And I said to the group, we can do better than that, you know. So we started. So that, I think, got me started with that aspect of philosophy. Another thing was Pat Churchland has been a wonderful influence, and I've known her from maybe 15 years or 20 years. I had a memory that seems to be a false memory. That in the beginning of her book, she talked about navigation and moving through space being a fundamental thing that animals do. And I looked back and I can't find it, and I've asked her about it, but I early on read her very famous book, Neurophilosophy, from 1990, and that was very inspirational. And I was very pleased after that to get to know her, and I know you know her also. And so I can't put a stamp on what got me started in thinking about it, but
does it motivate you still? The teaching is one motivation. Are you also motivated by? Yeah.
No, I've got this long manuscript kind of thing of trying to tie a lot of these ideas together, and hopefully it'll see the light of day someday. But
so at the start, when you were really interested in trying to understand who you are and emotion and so on, do you think have you gotten
yeah, I get ideas about emotion, and they fit in with this broader thing about consciousness. It would take me a while to lay that out.
Yeah, maybe another time, but I just wonder if it's if you found some answers or how that's progressed. But it sounds like you have.
Well, I don't know if answers. The field, neuroscience of emotion, is a difficult field. I mean, they seem to come together about 10 or 15 years ago, and now they spread apart again, like a consensus, but it's not there. But I have some ideas that, as an outsider, which I am.
Well, I hope you publish them or put them somewhere that we can read. And also, I hope you continue to document the downstate times. I know you've done a lot of interviews with Jim Runk, and I'll link to those and stuff.
We have Andre Fenton and I did an over-hour interview with Jim a couple of years ago that was really helpful.
I think also, that's very important.
Apparently read some of my blog posts, and that was a period of life that I really enjoyed it. And
they're actually really interesting. I'm going to link to them
but I want to start up again. I've got a whole list of topics that I never quite
good. Because at some point I was looking at your blog. I think that was a few years ago, and it doesn't seem up yet, but I hope you put it back up.
Oh, it's there, I think, but it's not. Nothing,
Oh, I couldn't get it, but anyway, I'm going to let you go now because you need a rest.
One interesting thing about the blog, you can't stop. So there were two blogs. There was a personal blog, which just got me going. And then I was part of a group sponsored by the Society of Neuroscience bloggers about
brain facts.
Yeah, but that doesn't go on anymore. They stopped it. What was interesting about that was I asked, what am I supposed to do? What do you want? And they said, well, make these posts sort of where, say, an advanced high school or college student interested in neuroscience could understand them. And that was a challenge.
Yeah.
To make it interesting, not dumbed down, but understand sort of a little bit what you're doing. I wrote my own one. It was sort of a fellow neuroscientist. I wasn't giving much background, but to the Brain Facts blog, I found it was a fun challenge to try to make it, to introduce it in a way that a bright college student could be interested and follow along.
Yeah, I find it it feels light, but you actually learn a lot. And because you relate it to things like a New York Times story or Memento, the movie, or golf, or, and I think that, you kind of sneak in the learning in a way. So it's good.
Thank you.
I just want to say thanks. It's great. As you know, I think that this time period is really important. And I think we're learning something about the relation of mind and environment and the body from it all that is a very big deal. So on that level too, I say thanks for all the work that you've all done. And I wish you the best.
Thank you for inviting me. It's been fun. Hope it made some sense.
It was wonderful. Is there anything you wanted to say that, before we go that I haven't asked, or I know we've been talking.
Reminded of the movie ending, which is, hi, Mom.
That's perfect. Let's leave it there.
No, nothing to say.
All right. I'll stop recording.